So let's talk about the PCA. The PCA stands for Principal Component Analysis. Without going into the mathematical details of the PCA, we will try to limit ourselves just to the interpretation of the PCA plot with reference to the RNA-seq data. But before discussing the PCA with reference to the RNA-seq data, let's try to understand its basic underlying principle. Let's assume for a moment you have one bucket full of the fruits, having the apples, grapes, bananas, orange, and a pineapple. All of these fruits are mixed in the bucket. But you know the features of each fruit. You know it very well that the apples are red in color, while the grapes are purple in color. Similarly, you also know it very well the pineapple is in yellow and the green, while the bananas are basically having a yellow peel. With this information, you are asked to separate all of these fruits on the basis of their features. So what should we do? By looking at the features of the fruits, you will start to put them at one place. What does it mean? It means that you will collect all of the oranges from the bucket and you will put them on one specific position. Similarly, you will take all of the apples based upon the features and then you will put them on one place. And likewise, you will put the bananas and the grapes on the separate positions. Now keep it into mind that you did all of the separation by keeping the common features of the fruit in view. All of the oranges are alike and likewise all of the apples are alike. And that's why you put them in a separate position. By the way, what you did right now is basically the principal component analysis. You separated the stuff from one another based upon the common features in them. Likewise, in case of the RNA-seq analysis, we are going to apply the principal component analysis. Let's assume for a moment that we have two different cell lines, A and the B. We are going to take A as a control, where we are going to give a treatment of a certain drug to the B cell lines. And we want to compare the differential expression of the gene between them. For A cell lines, we took 4 samples, and similarly for B cell lines, we also took the 4 samples. After applying the DE-seq function on this data, we built this principal component analysis plot. Here you can see that this is a 2-dimensional plot, having an x-axis and the y-axis. On an x-axis, there is PC1, while on a y-axis, there is going to be PC2. Now, what is going to be the interpretation of this plot? Now, this plot is letting us know that similar type of the cells are showing a similar type of the behavior. Like here you can see that all of the A cell lines are going to be clustered at one particular position. And similarly, all of the B cell lines are going to be clustered at one particular position on the PCA plot. But what about this A2? The A2 is not going to be present with the cluster 1 and the cluster 2 on the plot. This is going to be present at a very different position on the plot. This A2 could be potentially an outlier. And what does it mean? It means that the gene expression data in an A2 will be different as compared to the cluster 1 and the cluster 2 which is present on the plot. Now we need to consider it very carefully. It means that we need to redo all of the analysis for the A2, or we need to eliminate A2 from the rest of the analysis. Similarly, we could also have this type of the PCA plot for our analysis. Here you can see that not all of the A's and the B's are present on a separate different positions on the plot. Rather than they are intermixed with one another. But the one interesting thing which you can note it down here, that the A1 is present along with the B1, while the A2 is present along with the B2. And similarly, A3 is present along with B3 and A4 is present along B4. What does it telling us? This is telling us that there is a considerable similarity between A1 and B1, and similarly, there is also a considerable similarity between A2 and B2. But A1 and the A2 are quite different from one another. We hope so that with this discussion, you will have a good idea that how you can interpret the PCA plot in case of the RNA-seq data. In the next video tutorial, we will let you know that how you can build the PCA plot by yourself in RStudio.